Our next speaker is Kyle Breault. Kyle is from the Department of Biological Sciences. The title of Kyle's talk is Tiny Insects, Big Impact, DNA Analysis of Caddisfly Diets. Thank you, Kyle.
Raise your hand if you ate meat today. Fruit, vegetable. All right, that was easy. It's not hard to figure out what humans eat because we can just ask. But biologists can't do the same thing for animals. Sure, sometimes it's easy. We can see a lion devour a gazelle. But something like an aquatic insect that lives in freshwater ecosystems, this can be a little bit more challenging to answer. Yet biologists are obligated to do so. Because if we want to conserve and protect any organism or ecosystem, it is a prerequisite that we understand their food web dynamics, or who eats who and what eats what. In the case of aquatic insects, this is particularly important because they form the foundation of our freshwater ecosystems. And as our waterways become warmer, drier, and more polluted, we need to know how insect diets change over time so we know how they're responding to these negative stressors. That way we can make informed conservation decisions. But unfortunately, it can be really hard to evaluate what aquatic insects are eating. Take, for example, the Pycnopsyche caddisfly. The Pycnopsyche caddisfly falls into a feeding regime that aquatic biologists call shredding insects. This means that they eat dead leaves that fall into the water. And these leaves are going to be colonized by a diverse assemblage of bacteria and fungus, both of which are vital for any shredding insect's nutrition and health. But that's the major extent that we understand about the Pycnopsyche caddisfly and honestly other shredding insect diets. The problem is, if we want to know what they're eating, we have to break open their stomach contents and look under a microscope. But us biologists have a really hard time knowing what species of plant or fungus we're looking at because it's been eaten, it's shredded up, and it's half digested. So my solution is to use whole genome shotgun sequencing on the Pycnopsyche caddisfly's stomach contents. And for me, my workflow for the whole genome shotgun sequencing can be broken down into 3 major steps. First of all, I'm going to utilize some scientific wizardry to isolate the DNA from the plant, bacterial, and fungal cells that I find in the Pycnopsyche caddisfly stomach. Secondly, I will do DNA sequencing to get the unique genetic code for each organism present in their guts. Finally, I will compare the genetic codes I've compiled to the genetic codes of known species and match them. So this genetic code is this species, and this genetic code is this species. My hope is that my research can be used by other aquatic biologists in the future so we can better understand these insect diets, who knows, maybe even better than we understand the diet of a lion. Thank you.